Assalamu alaikum and hello everyone. My name is Muhammad Farhan Tanvir, and today my group will present a presentation on monoclonal antibodies therapy. Let's get started. So, monoclonal antibodies are often abbreviated as MAPs, and which are laboratory produced immune proteins. They are designed to bind to one specific target molecule, which we call an antigen. In our natural immune system, B cells produce antibodies to fight infections. However, those are usually polyclonal, meaning they are a mixture of different antibodies. Monoclonal antibodies are different. They are identical copies produced from a single B cell's clone. Because of this, they are highly specific and they can precisely target a particular receptor or protein in the body. Today, monoclonal antibodies are widely used in cancer treatment, autoimmune diseases, and often in infectious diseases. So, next slide. So now, why is targeted therapy so important? Traditional treatments like chemotherapy affect rapidly dividing cells, but they cannot clearly distinguish between cancer cells and healthy cells. This is why patients often experience serious side effects such as hair loss, nausea, and monoclonal antibodies work differently. They are designed to recognize specific receptors. For example, in certain breast cancers, cancer cells overexpress a protein called HER2. A monoclonal antibody can specifically bind to that HER2 receptor and block its activity. So, targeted therapy improves both safety and effectiveness, which is why monoclonal antibodies are considered a major breakthrough in modern medicine. Next slide. Okay, so historical development hybridoma technology in 1975. Traditional, okay, let me see. Okay, this is the slide. Okay, here's two scientists, Georges Kohler and Caesar Milstein, developed a technique called hybridoma technology. This discovery later earned the Nobel Prize. In this method, a B cell from mouse that produces a specific antibody is fused with a myeloma cell, which is a type of cancer cell that can divide indefinitely. The result is a hybrid cell we called hybridoma. This hybridoma can both survive for a long time and continuously produce one specific antibody. Before this discovery, scientists could not produce large quantities of identical antibodies. Hybridoma technology made large-scale, consistent production possible, which opened the door for therapeutic applicators. Next slide. However, Early, mon- early monoclonal antibodies had a major problem. They were completely derived from mice, which means they were murine antibodies, which injected into humans. The immune system recognized them as foreign and sometimes caused immune reactions. To solve this problem, scientists use genetic engineering techniques to gradually increase the human component in antibodies. First came chimeric antibodies, which are partly mouse and partly human. Then humanized antibodies, which are mostly human with only small mouse regions. Finally, fully human antibodies were developed. This evolution significantly reduced immunogenicity and improved safety for patients. So, the journey from murine to fully human antibodies show how biotechnology continuously improves therapeutic design to make treatments safer and more effective. To conclude my part, monoclonal antibodies are not just another class of drugs, they represent a shift toward precision medicine. Thank you. Now you will pass the presentation to Afik, who will explain the production process and mechanism of action.
Okay, Assalamualaikum and Akmal. My name is Afi Iskandar bin Abdul Rashid. Now, come to my part, which is I will explain about the recombinant production process. of DNA. So what's the first step? The first step is the antibody gene, which has a heavy chains and light chains are identified, and it will be inserted into the expression vectors. So the expression vectors will be introduced into host cells, which is acts as a replication of the to for the recombinant DNA. So the cell will start producing the monoclonal antibodies. This production will enable a large scale, which means that we can produce a huge amount of the monoclonal antibodies. So this modern production will be used recombinant DNA technologies in nowadays, such as like we use the bacteriophage as the host cell, which can produce a lot of insulins. So that's one of the example of recombinant DNA. Okay, now we move into the second part, which is the role of the CHO cells and the bioreactors. So the first one, which we must know what is a CHO. So what is the CHO? CHO is a Chinese hamster ovary, which is cells commonly used in the bioreactors in recombinant DNA. So the second one, you can provide a correct protein folding and the glycosylation. That's why we choose CHO. So the third one, we can culture it in large bioreactors. The fourth one, why we choose what's the role of the CHO, which is yes, control pH, temperature, oxygen, and nutrients. And the last one, you can allow industrial large scale antibody production, which is easy to use in large amount of the uh, CHO cells, industrial space. Okay, now come to my part, the third slide, which is the purification and the quality control of the recombinant DNA. So the first one, after we've produced antibodies, so antibody must be purified. Why we need to purify it? To avoid the microbial growth in that antibodies, so it won't spoil it. So for example, like protein A affinity chromatography use. So when we use it, after we produce an antibody, so we need to remove the impurities and the contaminant in the antibodies. So the quality test ensure the safety and the stability of the antibodies. So the second part is that we must to follow the GMP, good manufacturing practice, to uh, to ensure that the production already follow the procedure and every SOP which is already stated in the work. So the last one is the safety and the purity are critical for biologics. Why is it? Why is it? Why is it crucial? Because in production, so we have to ensure everything must be safety, efficacy, and quality. So this next part is the mechanism of action of the protein of the antibody. Okay, the first one is the receptor blockade. What is it? So this receptor, it will block signaling pathway in disease cells. So the second one is the ADCC. What is ADCC? Antibody is an antibody independent cellular cytotoxicity. So the immune cell will destroy this antibody bound target cells. So the third one is a CDC, which is the complement dependent cytotoxicity. It can activate the complement element and will cause the cell lysis. So the fourth one is the immune checkpoint inhibition. So it will boost the immune response in our body against the cancer cells, which is can cure the certain part of the cancer cells. So these technologies have a different antibodies which can have different mechanism.
Assalamualaikum warahmatullahi wabarakatuh. Ramadan Mubarak to all. As name Muhammad Nazmul Hussain. Let's start with one of their biggest success stories, oncology. You have probably heard of targeted cancer therapies, and monoclonal antibodies are as a prime example. Take breast cancer, for instance. For patients with a specific type, the HER2 positive breast cancer, antibodies can target that HER2 proteins on the cancer cell, blocking its growth. Similarly, the lymphomas antibodies targeted the CD20 marker on cancerous B cell, This approach has dramatically improved survival rates for these patients. The key here is selectively. Unlike traditional chemotherapy, which can harm rapidly dividing healthy cells like those in your hair follicles and gut, these antibodies are like guided missiles. They are far more selective, which leads to significantly reduced damage to healthy tissues. This remarked as a major breakthrough in cancer therapy. So move to next slide in autoimmune and infection diseases. But the power isn't limited to cancer. In this slide shows their expanding role. In autoimmune diseases like rheumatoid arthritis, the immune system mistakenly attacks the body. Here, monoclonal antibodies can act as peacekeepers. They targeted and neutralize inflammatory messengers, like TNF alpha, providing relief from chronic inflammation and joint damage. More recently, they played a critical role in the COVID 19 pandemic as neutralizing antibodies. Giving high risk patients a direct boost of defense, this really highlights how this technology is expanding beyond cancer to fight a wide range of diseases. So we have some challenges and limitations. Now it's important to acknowledge that these revolutionary tools isn't without limitation. So as we see on this slide, there are significant challenges. First, these are complex drugs to manufacture, leading to a high production cost. They are also large molecules, so they can't be taken as a pill. They require injection or infusion because they are proteins. The body can sometimes recognize them as a foreign, creating a risk of immunogenicity, where the immune system attacks at the therapy itself. And just like with antibodies, cancer cell can find ways to become resistant to these drugs over time. These factors, especially the high cost, contributes to limited accessibility for patients in low-income countries, creating a disparity in healthcare. The message here is clear. Innovation is still needed. Here is our future innovations, and that innovation is precisely what's happening, which brings us to the future on this slide. So, scientists are engineering entirely new generations of these drugs. We have bispecific antibodies, which are smart enough to grab a cancer cell with one arm and an immune cell with the other. So, bringing the two together to fight. Then there are antibody drug conjugates. Think of this as a Trojan horse. So it's an antibody with a powerful chemotherapy drug attached. So it delivers the toxic payload directly to the cancer cell, minimizing the side effect. We are also seeing AI assisted designed to create better, more effective antibodies, and the growth of biosimilars, which are like generic versions that will help lower cost and improve access. This all this is all moving us toward a future personalized biologics, where therapies are tailored to the individual patients. So we are reaching in conclusion. Monoclonal antibodies have fundamentally changed modern medicine. Their ability to be highly specific and targeted has had a powerful impact, especially in cancer and autoimmune disease. And as we have seen with continuous innovation, we are constantly improving both their safety and their accessibility. What we started as a laboratory discovery has truly become a life saving therapy for millions around the world. And finally, thank you, everyone.